eat. Well, that means they gotta find if it's a prey animal, they have to protect themselves from the predators. If it's a predator, they have to find the prey animal so that they can eat and survive. All of this stuff requires their special senses. If we look at the senses that are out there are 5 that are considered the general senses, and then there are the special senses, and there's also 5 of those. The general senses include visceral sensation. That means inside your abdomen. Anybody ever have cramps before? I mean, talking to women, well, except for Diego and me, we don't. But you guys have had cramps, right? That's visceral sensation, okay? Tactile sense means the sense of touch. Everything that has nerve endings in their fingers or whatever, they can feel touch. You may not know what it is, but you can feel something. Temperature sensation is another general sense, the ability to sense what the temperature in the environment is, or when you touch something. Pain or nociception is another of the general senses. You guys are going to learn more about pain because pain and pain control is one entire section on your national board exam. Okay, on your VTNE. You've got to do a whole section on just pain and pain control. And then proprioception, and you already know what that is because we've talked about that in neurology. The ability to know where you are in space or spatial relationships. Like right now, you guys all could close your eyes and tell me if you're sitting down or standing up, right? Because you can sense that. I can tell you if I close my eyes that I'm standing up because my brain knows that. Then we get to the special senses. The special senses include the gustatory sense or taste, and then the sense of smell, okay? And then we've got sight, the ability to see, hearing, and then balance. The ability to stand up and not fall over is a really complex sense in our bodies. So let's start with the general senses. The first, visceral sensation. All of you that have had cramps in the past, can you tell where it actually hurts? No. You just know it's somewhere in there. But where exactly is it? That's the thing with visceral sensation. We can't really figure it out. We know that if we're hungry, it's somewhere in there, but exactly where. If we're thirsty, I know somehow what that is. And then the same thing with the cramps, or if you eat too much. Anybody ever done that and regretted it the next day? Or even an hour later? Tactile sense, or the sense of touch, is a pressure type sense. When you put pressure on something or something puts pressure on you, it activates specific nerve endings. These nerve endings are in something called Meissner corpuscles or Pacinian corpuscles. And if you look at this thing, this is a Pacinian corpuscle, and right in the center is the nerve. Now, if I put a little bit of pressure out here on these layers out here, that's only going to transmit a little bit to here. If I put more pressure, so it'll go to here, now it's going to feel like there's more pressure there. So these are the kind of senses that change based on the amount or the degree of stimulus. Okay? So we've got light, head touch, we've got deep pressure, vibration, and then even hair motion. Now, we're not so good at the hair motion thing. But have you guys ever watched the rats when they're trying to move around, or a mouse going through a little hole, or the ferret? How do you think they can figure that out? Their whiskers. They can feel what's coming at them. There are some animals that are virtually blind visually, but because they've got good sensation, they can tell where they're at. Even the bird, she can tell, or he can tell us what's going on when you touch his feathers, even if he can't see you touch his feathers. There's not many of us that could do that by just touching the hair. You can feel it, but not nearly to the degree that they can. The next of the general senses is the ability to sense temperature. Now, there's different kinds of temperature sensation. The first is superficial, and that generally is because of nerve fibers, nerve endings that are close to the skin, and it tells what's happening. You can tell when it gets cold outside versus hot outside, just because of the sensation on your skin. But then we have the other sense that's really important, and that's core temperature and the regulation of that core temperature. That regulation is up in the area of the hypothalamus, okay, way up inside there to help us maintain temperature at a specific level. As the fact that we are endotherms means that somewhere in our brain it tells us how we can maintain temperature at a certain level or within a certain range, okay? Most mammals are like that. Obviously, ectotherms are not like that. And we'll talk more about ectotherms when we get into the next section. The sensors that are for temperature regulation are free nerve endings close to the skin, or another kind of sensor arrangement called a ruffinian corpuscle. The next of the general senses is pain or nociception. The sense of pain is different for different levels, okay? Superficial pain is pretty simple, okay? You step on a tack, you know you stepped on a tack, right? And you know exactly where that is. That's superficial pain. Deep pain, on the other hand, is not recruited until something really serious happens. It means that you've broken a bone or something. It's completely different, okay? When it comes to being able to determine superficial pain and deep pain, what do you have to do in a physical exam? You have to hurt them. You have to try and hurt the patient. Well, does that make sense? Does that seem weird? No. You have to do that, especially if there's a problem like a spinal cord injury. Now, if an animal has superficial pain, do you have to worry about deep pain? No, because you know they got that. Because they lose it in order. They lose their ability to feel pain from superficial pain, and the last thing that's lost is deep pain. So if I'm working with an animal that I think has lost pain, I literally have to try and hurt them to make sure I know for sure if it's happened. I've been in this profession a long time, and I've been a surgeon for a long time, and I would get phone calls from referring veterinarians that, usually it's dachshunds, little chondrodystrophic breeds that have spinal cord problems, especially when they rupture a disc. And they'll call me up and say, I've got a little dachshund, it's ruptured its disc, and I'm sending it over to you for surgery. And I would ask, tell me what your physical findings are. And they tell this, 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 and this. And they would always say that it feels pain. And I would ask them, how do you know if it feels pain? Well, when I pinch its toes, he pulls his foot back. That is not a pain response. That is a reflex. In order for it to be determined as pain, they have to respond consciously to that. That means they have to feel it. And the only way that you know if they feel it is that they have to acknowledge it. They have to try, they have to bite at you, they have to do something to try and stop the source of the pain. Well, if an animal's just pulling its leg back, that's just a reflex. So you have to go further. I literally have gone in with needles, perforated in through the skin, and scraped the periosteum on the bone to try to find out if they feel it. And if they can't feel that, there's probably a good chance their spinal cord is destroyed. And if that's the case, the prognosis just went into the toilet. Then these people would say, well, should I send it for surgery? So you got to talk to the client, because the surgery may not do a thing. If the spinal cord is destroyed, it's not coming back. So being able to understand the concept of superficial and deep pain is critical in what we do. And yes, you got to try to hurt them. Now, you don't have to try and hurt them if you can see that a slightest little impulse is going to cause them to hurt. Like if you pinch their toe and they turn around and try to bite you, or they cry out, you don't have to go any further, right? Now, the visceral pain, we've already kind of talked about. That's the cramps, okay? That's not just a sensation, a tactile sense, but now it can be a painful response. Sensors are free nerve endings, again, that are really easily stimulated. The behavioral adaptations, however, are completely different. For example, how do you know if a cat hurts? Does that mean they hurt? If a cat's laying there meowing, does that mean it's in pain? Maybe. What if they're cold? What if they're nervous? Do they hide? So does that mean they hurt? Anybody ever figured out a dog? Anybody here got Siberian Huskies? Would win with the dog world? Versus a pit bull. The same sensation on a pit bull would destroy the Siberian Husky. I had a pit bull come into me when I was in private practice years ago. He had just been hit by a car. His head, his skull was caved in right here, and every time he breathed, his head was doing this. Okay? Because the bones were busted. The dog wanted to play. He came in and he was wagging his tail and was acting great. Had another dog, a Siberian Husky, came in that tore his toenail, and you thought he was going to die. He was screaming out every time you touched him. Didn't matter if you touched his toe or not. He thought he was dying. So what I'm talking about now is the behavioral adaptations for specific types of animals. Cats versus dogs. People versus animals. Have you guys ever seen, I'm not picking on women specifically, but it is kind of funny. Have you ever seen a woman that's gone in for an ovariohysterectomy? What do they feel like after the surgery? They're laying on their back, they're thinking that they're going to die for probably 2 or 3 days before they even consider moving. Now you do the dog. What do they like the next? Within a few hours. It's the same surgery. So why are they so much different from people? Because of the different adaptations. And that has a lot to do with the kind of animal. All right, proprioception. We've already talked about proprioception in neurology, so we know about that. The ability to recognize where you are in space, or where your limbs are in space. And then the ability to know. The ability to walk in a straight line and not fall over or not deviate from one side to the other side is really unique, as long as you've got some place to recognize where you are. Okay? Anybody ever been in the deep forest and been told to walk in a straight line? Think you can do it? Not a chance in Hades. If you don't have some point of reference, your body's just going to start going in circles. They've done this. They've done this experiment. They took people into a forest. They told them to start walking in a straight line. These people thought, no, piece of cake. Then they put a GPS on them, and then they showed them what their pathway was on a computer. Some of them had complete circles within a matter of 100 yards. Because they couldn't tell. But they didn't fall over. They just, one step was a little longer than the other step or something. They were able to stay up and stay conscious, okay? Because they had proprioception. The receptors or the sensors for proprioception are stretch receptors usually that are found in muscles, that are found in tendons, ligaments, all over your body. And what they do is they send signals to the brain constantly to say where things are, where you are, what position you're in. It's really a unique thing to have, but it's also one of the first things that's lost in the body when the spinal cord is impinged. Even before pain, proprioception is one of the first things that is lost with any minute pressure on the spinal cord. Okay? All right, now let's get to the special senses. We're talking special senses. The first one is the gustatory sense, the ability to taste. Now, the taste receptors are in the tongue, okay? Some animals have them in other places in their oral cavities. But it's generally in the tongue, and these are chemoreceptors. What these chemoreceptors do is they have little hair-like projections on them. And they kind of stick out when whatever goes into your mouth becomes solubilized, and that chemical then touches the hair, it sends signals. The signals that are sent include sweetness or sour, salty or bitter, or a combination of any or one of the above. Okay? That's how you have specific tastes. Now, what's really interesting is the sense of taste and the sense of olfaction are closely associated. The same chemicals that will stimulate your taste buds are the same chemicals that stimulate your olfactory organ, your smell, sense of smell. Okay? And they essentially work the same way, except one is the opening to the GI tract, the other is the opening to the respiratory tract. If we look at the sense of smell or olfaction, it's completely different depending on the animal. Okay? Some animals have what's called a macrosmatic area, or area for olfaction. Their olfactory bulbs are completely different size. Some of them are microsmatic, very, very small. And some of them don't have any. Now, if you look at that coyote up there, what do you think his sense of smell is? Extraordinary. Not as good as a bloodhound, but it's still pretty impressive. But then that primate, that mandrill up there, that's about equal. We can smell things, but not nearly to the degree as a canine. And then anosmatic was the walrus. They can't smell anything. They sure have a good sensation of touch, tactile sense, but they can't smell a thing. Okay? And I'm pretty sure that the sense of smell in birds is different, especially vultures. How can you stick your head inside of a rotting body, carcass, and sit there and eat it? That drive us nuts. So there's got to be some differences. The sensors are in the olfactory epithelium, and that's in the area in the back of the respiratory tract in the nasal cavity. And it's very closely associated with the cribriform plate that goes into the brain. On the opposite side of the cribriform plate are the olfactory bulbs, or cranial nerve number 1. So it's not a very long distance. The chemicals that you inhale, or the odors that you inhale, when they get into the moisture of your nose, it becomes soluble, and those soluble chemicals stimulate the hair cells that are in your olfactory epithelium, similar to the taste buds in your tongue. So they work the same. Okay?
So it was in the back.
Back of your nasal cavities. All right. Now we get to vision. And the ability to see things, and how this works. And this is really, really impressive. Okay? But unfortunately, you got to know some physics. Okay? Now, I know physics is one of those words that scares a lot of people, like math. But it's just physics, okay? Not a big thing to worry about. When we're looking at optical physics, there's things you have to understand. The first one is this thing called the refractive index. And what that means is that light will transmit through a transparent substance. But as it goes through that transparent substance, it slows down. It's not the same as light just traveling through air. Okay? So what that means is, and you guys know how fast light travels, right?
Speed of light.
Guess what that is? What the sign is for speed of light? C.
C squared.
C. No.
Well, C squared is light.
No.
C?
C is light speed. C squared is light speed
Squared.
squared.
But the C is light.
Yes, the C is light. E is equal to mc squared. C is the speed of light. So the speed of light in air is different than the speed of light through a transparent substance. And that ratio is the refractive index. Okay? Now that's really important because we're going to get to some things that are transparent the light has to go through. Then the next thing is refraction. Refraction means that light will bend as it transmits through a transparent substance. Okay? And as it goes through that, it's going to come out at a perpendicular to the point of exit. Okay? So if you look at these lenses, we see a convex lens here. Light comes through perpendicular, but then when it exits, because there's a curve there, it leaves at a 90-degree angle. So it's bent that light. And that's how they figured out how to help us see better with glasses. Because it changes the point of refraction so it'll hit our retinas in the right location. Okay? Everybody understands who's nearsighted in here besides me? Okay, you know what that means? It means your eyeball's too long. If you're farsighted, your eyeball's too short. And so the light, when it goes through the lens, doesn't focus on the retina. That's why we put glasses in front of our eyes. Okay? Or contact lenses. Same thing. Now, the convex lens, the lens that is bent like this, it always causes the light beam to converge to a certain point. Okay? Whereas if it's a concave lens, it causes the light waves to bend and diverge from each other. Okay? So by knowing what the refractive index is, and what that means, and what refraction is, we can start changing where light goes based on the shape of the lens we put in the pathway of the light. Okay? Now that doesn't just mean people will wear glasses. Okay? So if we look at these optical physics and can go on with this production of the image on a different surface, anytime you have a convex lens, by the time the image is back to where it's supposed to be, it's turned upside down and backwards. And guess what? Each of you has inside of your eyeball a convex lens. So that means everything you see that your brain receives is upside down and backwards. But your brain puts it back in the right order. That's what your brain gets to do. But you can see this if you just showed things on a regular projector and just showed it just through the convex lens. When you look at it, it's upside down and it's turned backwards. Okay? Now, this interpretation, as long as everything's going okay, it's going to put this right side up and turned around again. Okay? Everybody got it? All right, so now let's look at this concept of sight. Think of the eye and a digital camera as kind of analogies. The lens cover that you put over the surface of the camera lens, that's just like your eyelids, okay? Same kind of an idea. Then, the window, the first lens, or the first covering over the lens of the camera, they sometimes just call them a shield or a filter sometimes. But that little lens or that window is like the cornea of your eye. Now, the difference though, in cameras, that cover is usually flat. Is the cornea of your eye flat? No, it's curved. But it's not convex. It's two layers that are parallel, but it's still curved. Okay? Then the next thing we have is the diaphragm of the camera. That's what lets light in. And then you can change how much light gets in, right? By just changing the diaphragm on the camera. That's like the iris of your eye. Okay? Then the lens of the camera, the actual lens, is like the lens in your eyeball. The light detectors in the digital camera is the same, the retina in the back of your eye. And then the last thing is the transmission cable that takes that image to the little screen that you can see. That's basically the optic nerve in the eyeball. Okay? So now let's look at the anatomy. We're going to first start talking about the globe of the eye itself. The first thing we have to look at is the outer layer, the thing that holds it all together. The fibrous outer layer of the eye, and in that image, it's the white part that you can see goes all the way around. That's called the sclera. And the sclera is really tough. And if you look at your classmate's eyes, you can see it. You can see the white part of your eyeball. Okay? That's the sclera. The sclera is covered by a loose tissue called the conjunctiva. The next part of this outer surface is the cornea itself. The cornea is confluent with the sclera. But you can see right there, it changes from being white, tough, to perfectly clear. Okay? Now the thing you guys have to understand, this is a three-dimensional structure, not two-dimensional like this picture. So that means this part right here comes out and goes around up to there. Okay, so think of this in a three-dimensional pattern. Now, the point where the white sclera and the cornea come together is called the limbus. Okay? So the limbus is 360 degrees. You can look at your classmate, you can see it all the way around. Okay? Now, the next layer to look at, not the outer layer, which is the sclera and the cornea, now we're going to look at the vascular layer. The vascular layer of the eye is called the uvea. Okay? What do you think you call it if it's the uvea gets inflamed? Oh. Ever wonder where that word came from? The vascular layer of the eye is now inflamed. The uvea has three parts. Okay? The first part is the choroid. The choroid is the vascular supply way in the back of the eye. You can see it, kind of that darker red layer. Okay? That's the choroid. And remember, it's 360 degrees, 3 dimensions. Okay? Now, inside the uvea, the choroid specifically, there is a certain structure that some animals have and some animals don't. Okay? And that thing is called the tapetum. And the tapetum has a special purpose, and that is to reflect light back from the vascular layer back to the retina. So that means animals that have a tapetum, light goes through, hits the retina, goes through the retina, into the tapetum, reflects back to the retina. So what does that make those animals able to do? See better at dark. Guess what animals don't have one of those? People don't have them. And pigs don't have one, okay? Those are things that are really important. So if you guys are driving down the road at night, and all of a sudden you see two little spots way up in front of you, what do you think that is? That's the light reflecting off the tapetum of an animal out there somewhere. Rabbits and deer have them. If you shine a light on a person, or if you're driving down the road at night, and a person was standing in the road, would you see a light reflect back? No. So whenever you see that in movies, people's eyes shining back, no, that doesn't work. That's not real, okay? Because we don't have that. Now, the next layer, or next part of the uvea is the iris itself. And the iris is the colored part of your eyeball, okay? So if you look at your neighbor's eyes and they have blue eyes, that means their tapetum, not tapetum, their iris area, part of the vascular area, is blue. Get her back up on her perch.
Going back.
All right, so that iris also has something special in it. It has muscles, because that's what allows it to change its shape or its size. So the pupil size gets bigger or gets smaller, depending on the light that's in there. Put her up there, you can't make her lower. Put your arm down. Now, right now, put your arm down lower than the perch. Now, push upwards. Push her into the perch. Push her into the perch. Now start to pull your arm away. No, not that way. Push her into the perch. Now you're getting her up to your shoulder. You guys need to figure out how to show Sawzall know who the boss is. Step up. Now get up there and stay there. All right. The next thing that we have to talk about, or part of the uvea, is the ciliary body. The ciliary body is a portion of the uvea that's right at the junction of the iris and the uvea. The ciliary body is really, really important because it has cells in there that produce fluid, kind of a watery fluid that flows into the eye itself. We'll talk about where in a minute. The ciliary body also has muscle inside of it, smooth muscle, which means what? Involuntary. It means we can't control it unless you're a bird. Because birds can control theirs. Skeletal muscle, okay? And then there are suspensory ligaments that go from the ciliary body to the lens and attach to the lens, 360 degrees around on that convex lens, okay? So as the muscles contract, they put tension on those ligaments and it changes the shape of the lens. Well, what does that do? Changes the focus. So that's called lens accommodation, or the concept of accommodating. Now, as you guys get older, you're going to get hard lenses. The lenses in your eyes are going to start hardening. That means they're not going to change shape based on the pull of the ciliary body. You know what happens, what you guys have to do when that happens? No, get reading glasses. Because that's why we can't see things as much. We have to do this, play the trombone when you're trying to read something. Okay? Because your lenses don't change shape anymore. Have you guys ever watched a horse? When they try to see something, what do? Because their lenses don't accommodate very well. They have to move their head. And And they move their head pretty quick. And if they move their head really quick and you're in the way, you're going to get hurt. Because they can't accommodate nearly as well as other animals or younger animals. All right, so now we've gotten through the outer layer, the fibrous layer, the sclera and the cornea. We've gone through the uvea, the vascular layer, which includes the choroid, the iris, and the ciliary body. Now we're going to get to the next layer of the eye, and that's the retina. The retina of the eye is this kind of tan layer that you can see on top of the uvea right there. Okay? This is where all of the impulses are created when an image is reflected back or transmitted to the retina. Inside of the retina there are photoreceptor cells, and they have been classified into two types, the rods and the cones. Those two types of neural receptors are different in the kind of impulse that they're sensitive to. Okay, we'll get to that. After the rods and the cones, the photoreceptor cells, then we have something called a bipolar neuron. What does bipolar mean? Two ends, and they both receive and transmit. They receive the impulse from the photoreceptor cell, and then they transmit it to the ganglion cells. The ganglion cells have a really long axon. And those axons, all those ganglion cell axons, all leave the eyeball at one location. And that's what becomes the optic nerve that goes out of the eye to the brain. Okay? So if we look at these things, the rods are very, very sensitive to light and dark. And that means that that light that comes back from the tapetum is going to stimulate them again, because that's light and dark. The cones are very sensitive to color. Okay? So that means the different things that we see as different colors are stimulating primarily the cones. Now that doesn't mean the rods aren't there too, because you can tell things what color they are as long as the amount of light and dark is correct. Anybody ever try to figure out what color something is when it's just really dark outside? Can you tell what color it is? No. You've got to have enough light coming in to allow the cones to work properly. Okay? And speaking of that, what color is blue? But what color is it? Is your blue the same as my blue? How do you know? Maybe when I look at the sky, I see blue, but what I see as blue might be your orange. So you might be looking at the sky and it's just this bright orange color and you'll say, that's blue. Have you ever thought of that? It's kind of a weird concept. You see, and the reason I bring that up is because animals don't see colors the same way we do. They may not see the same kind of thing. They may have a completely different spectrum based on the visual spectrum. Okay? One of those things to think about. When you go, Now, the bipolar neurons, again, what they're going to do is they're going to, for example, there's the photoreceptor cell up there. There's the bipolar neuron. It's going to receive the impulse from the photoreceptor, take it down through, and deliver it to a ganglion cell. Now, when you look at that, that's the order they're in. So the photoreceptor is really close up there to the choroid, okay, where the tapetum is. It kind of makes sense, right? Then we have bipolar neurons, and then we have the ganglion cells way out here. The light hits the ganglion cells first. It has to travel through the ganglion cells before it can hit the photoreceptor cells. And then the impulse is sent back here, and then out to the optic nerve. Okay? All right. Any questions about the surfaces or the layers of the globe? We have the sclera and cornea, then we have the uvea, which consists of the choroid, the iris, and the ciliary body, and now we have the retina. Okay? There's other structures inside the eye, and the first one to talk about is the lens itself. The lens is either a spherical or a convex shape based on the tension that those ligaments from the ciliary body are putting on the lens. Okay? If they're relaxed, the lens is elliptical. It forms that convex shape. If they're contracted, the lens becomes more round, okay, like a ball. And that is that concept we talked about earlier of accommodation, or the ability to focus. Because remember refraction and refractive index and all that stuff we talked about? If you change the shape of the lens, the way the light comes through that lens is going to change, okay? Especially on the refraction side. All right, so now we've got sclera and cornea. We have the uvea, which is choroid, ciliary body, and iris. And now we have the retina. We've talked about the lens. So now let's talk about putting everything together. There are different compartments inside the eyeball. The first compartment, as you're traveling from outside to inside the eye, okay? After you've traversed the cornea, you get into something called the anterior chamber. This is one place where the word anterior is used in animals, okay? Because it is in front of. So anterior chamber is the chamber in the eye just past the cornea. So when you look inside your classmate's eye and see the iris, you're having to look through the cornea, through the anterior chamber before it even gets to the iris. Okay? The posterior chamber is the chamber that is between the iris here and the lens. So it's just this little area right there. This is the ciliary body. Okay? So that ciliary body has special cells in it that create what? Fluidy stuff. It's secreted out here. That fluid goes into the posterior chamber, flows through the posterior chamber, comes out through the pupil, and goes into the anterior chamber and fills this up with fluid. Now, those cells are continuously producing fluid. So what does that mean? It has to happen. They're continuously producing fluid as long as your eye is acting normal. Huh?
Would have to like go somewhere.
It has to go someplace. It has to drain out. Otherwise, your eyes will just keep getting bigger and bigger. Okay? So that fluid comes out, goes over here to the drainage angle of the eye, and flows out through little tiny vessels. And guess what that becomes? I knew you were going to say that. No, it's not tears. It flows into blood vessels. Where do blood vessels go? Back to the heart. It takes all that fluid back to your heart. Tears are something completely different. And every year I go through this, and every year someone says that tears come from the anterior chamber. Don't do that. They don't. They are not related. Okay? Completely different source. Tears and aqueous humor, or the fluid that's inside the eyeball. Okay? All right. Once you get on the other side of the lens, you've gone through the anterior chamber, through the posterior chamber, there's the lens sitting there. Caudal to or posterior to the lens, we have the vitreous compartment. And the vitreous compartment contains a vitreous humor. Up here is aqueous humor, or watery stuff. Back here, it's not watery. It's gelatinous. Okay, but it's clear. Hopefully you guys are seeing what's coming. It's clear so that light can transmit through it. A clear substance means that light's going to go through it slower than through air, right? That refractive index we talked about. So now you've got the cornea, you've got the aqueous humor, then you've got the lens, now you've got the vitreous humor, and the light has to travel through all of those to get to the retina. So that's where refractive index plays a big part. Any problem in any of those different areas can change what you see or how you see. Okay? That fluid again in the anterior chamber and posterior chamber comes from the ciliary body, and that's the fluid that provides the nutrients to the inner surface of the eye, especially the inner surface of the cornea. That cornea is not one cell layer thick, it's pretty thick. The inner layer gets its nutrition from the aqueous humor. Where do you think the nutrition to the outer layer comes from? Blood? Do you have blood vessels in your cornea? No. So that if you did, you'd have a problem. So where do you think the nutrition to the outer layer comes from? Think it toward. Well, that's on the inner surface. Huh? You really got me confused. Where does the nutrition to the outer surface of your cornea come from? Tears. The tears have to provide that nutrition. Otherwise, it dies, unless there's been damage to it. And we'll talk more about that later. All right, then we get to the muscles of the eye. And this is really cool. Anybody know who Marty Feldman was? Who's Marty Feldman? You guys ever watch Young Frankenstein? Igor? His eyes could go this way and this way. That's a really impressive talent, because the extrinsic muscles of the eye are skeletal muscle. So that means he could make his eyes go different directions just by learning that, training his eye muscles. Normally, our eye muscles work in concert with each other, because you got the same eye muscles on this eye as you do on this eye. Okay? If we look at those muscles, we have rectus muscles. What does rectus mean? Straight. They're straight muscles. Okay? We have the lateral rectus. The lateral rectus is a straight muscle. Guess where it is? On the lateral side of the eye. Then you have a medial rectus, which would be exactly the same position, but on the other side. So it's inside here. Okay? Then you have a dorsal rectus. The dorsal rectus is dorsally positioned. So here's lateral. On the dorsal surface, there's another straight muscle. And then we have a ventral rectus. Ventral rectus is going to be opposite of the dorsal rectus, so it would be down in here someplace. It would be hard to see there. So that means you have four muscles that are straight, that go back this direction. Now, if the muscle contracts, if my lateral rectus muscle retracts or contracts, what would happen to my eyeball? If my lateral rectus muscles contracted, my eye would turn this way, and I'd look over there. But if my lateral rectus on this eye contracts, what happens on this eye? My medial rectus has to contract. That way, both eyes go the same direction. Make sense? If I want to look down, my ventral rectus muscle contracts, but both of those contract at the same time. Dorsal, the same thing, because it's going the same direction. But when it comes to the lateral and medial rectus, they have to work in concert, but it's a different muscle. okay? The oblique muscles are also there. The oblique muscles are the dorsal oblique muscle, okay? Dorsal oblique muscle is, where's number 3? Right up there, okay? Now, it's dorsal and it's oblique, and it sits really close to the skull, okay? But it causes a different kind of reaction or action in the eye because of this thing called the trochlea. The trochlea is kind of like a pulley that changes the direction of the pull. So if the dorsal oblique muscle is up in this area and it's on the dorsal surface medially, and if that trochlea comes around and it makes it attach to the eyeball here on this side. So what would that do to my eyeball? Causes it to rotate. So the dorsal oblique muscle, when it contracts, it causes my eyeball to do this. Make sense? Then we have the ventral oblique. The ventral oblique is over here, and it attaches to the eyeball on this side. So when it contracts, it makes my eyeball do this, rotate outward. Okay? So now I can go left, right, up, down, and then I can rotate my eyeball. The other muscle that is real common in animals that humans do not have is one called the retractor bulbi muscle. The retractor bulbi muscle kind of surrounds the optic nerve, and it's inside or covered up by the rectus muscles. Okay? And if you look where the optic nerve is, that kind of surrounds them. And it attaches to the back of the eye. And when it contracts, it pulls the eyeball back into the head. What happens when the eyeball of an animal gets pulled back into the head? Especially a dog or a cat. Third eyelid comes up. That's the only way the third eyelid comes up, is when the retractor bulbi pulls the eyeball back. Okay? Except a bird, obviously. Because they can make their third eyelid come up just by wanting it to come. Okay? Why does a dog retract the bulbi work? Something hurts their eye. Something's irritating, so it pulls back. So the third eyelid will come up. Okay? And that brings us to the outside. Okay? The eyelids of the eye are called palpebra. There is a dorsal and a ventral palpebra. So the upper one is dorsal, ventral is lower. Where the eyelids meet, laterally and medially, are called canthi. So the lateral canthus is over here. The medial canthus is close to the nose. Where the canthi meet, that's a place where the conjunctiva also can attach. Now remember, we said that the conjunctiva is a covering for what layer? Which is what? Oh, that's inside. The sclera. The sclera is the white part. You see those tiny little blood vessels? That's the conjunctiva. The conjunctiva is where the blood vessels are. So here it is, and that conjunctiva stops right there, because it doesn't cover the cornea. It stops here, but then over here by the eyelid, that conjunctiva turns the corner and now covers the inner surface of the eyelid. Okay? Now, the third eyelid, nictitating membrane that some animals have, is also covered with conjunctiva. That conjunctiva comes from the eyeball way down under here. It reflects up on the third eyelid. It goes over the outer surface of the third eyelid, and then reflects back onto the eyelid, the outer eyelid. Okay? So it's very, very loose and movable when you're looking at an eyeball. Now, inside of that nictitating membrane, or the third eyelid, there is a gland called the nictitans gland. Very important because that's what helps to produce tears. That's only one of the tear glands. Okay? So that tear production, again, there is a lacrimal gland, okay? The lacrimal gland is on the lateral surface of the eye, dorsally. It's over here. But there's also one in the nictitamus gland. So there's two tear duct or tear glands for each eyeball. And they produce tears. And why are tears important? It's nutrients to the outer surface of the cornea and to prevent the eyeball from drying out. Tear drainage, though, has to go someplace because the tear ducts, the tear glands are producing tears. So it's got to go somewhere. If you come to the medial canthus, on the upper and lower portion of the eyelids, there's two little holes. Okay? Those two little holes are called the puncta. And the puncta are where the eyelids, or excuse me, where the tears drain. So, if you look at your classmates' eyelids real closely, look right here and right here, you're going to see two little openings. And you probably can see your own if you look in a mirror. Okay? The tears have to go through those holes. Then they go through little tiny canals. Okay? So here we have puncta here, puncta here. They're going to go through two little canals and they're going to meet over on the side. And they meet in something called the lacrimal sac, where all the tear just kind of pools in. And then that lacrimal sac gives rise to a little duct or a tube that goes from the lacrimal sac to the inside of your nose. That's called the nasal lacrimal duct. So, if you're crying, what also happens? Your nose runs, because the tears are coming out of your nose. Okay? What happens if one of those gets blocked? Huh? Well, just the duct, just the nasal lacrimal duct, which can be catheterized. If you find you can put a catheter into it. What happens if it blocks up? Tears are being produced. Can't get into your nose. It's going to overflow onto the inside surface of your eye. If you have a white dog that has a brown streak right there, that's tears that flow over, and that's called epiphora. Okay? Sometimes it's normal because there's just too much tear production and it flows over. If a nasal lacrimal duct is blocked, it'll flow over. But how would I find out if that's the problem? I'll put a catheter in the puncta up here and flush it, see if I can get stuff coming out of the nose. Or I'll put something in the tears that turns the tears a certain color. And then if I see that color coming out of the nose, that means I know it works. Anybody ever seen that done? They use fluorescein stain in the eyes, and you can see it come out of the nose if the tear duct is working. If it's not working, that might be why they have the epiphora. Make sense? All right, we'll stop there. And that means now you get to go into the lab and color eyeballs.
Woohoo!